students, I am Tulika Banerjee. Today, I bring you the next learning episode in BSc Forensic Science on an important unit of the paper titled Fingerprints, that is, importance of lip and ear print. In this lecture, you will be studying about lip and ear prints, their history and classification. In addition to it, you will also be studying about how they are developed and analyzed. Moreover, you will also study how they are important in the field of investigation and their validity in the court of law. We will wind up this lecture with a conclusion. Let us start our today's lecture with a brief look at what we will be learning today. First is introduction, then history of lip print, classification of lip print, development of lip print in crime detection, lip print in court of law. Then we have ear print analysis, validity of the ear print as a forensic evidence, and lastly the conclusion. Dear students, let us start with the introduction of this lecture. First is lip print. The study of lip prints is called as chiloscopy. An individual's lip prints are exclusive alike to fingerprints. Lip prints are rarely used in criminal cases as its creditability has not been resolutely established in our courts. There are two ways of possibly using lip prints. First, print pattern for identification, and second, chromatography to match lipstick marks. Identification plays a major role in any crime investigation. The shape of creases on the lips has distinct features like fingerprints. Chiloscopy is a forensic investigation method that deals with identification of humans depending upon the on-the-lips traces. Larger studies on lip print have been carried out by various scientists called as tool for human identification in both civil and criminal cases. The lip crease shape exists on the vermilion border of the lip, which is relatively movable, and lip prints may vary in form as per the pressure, direction and method used in making the print. Next is ear prints. The awakening of forensic interest in the description of ear prints and in identification by means of ear printing is relatively recent. It constitutes one more step in the interminable race between criminal methods and criminalistics science. It is even harder to find laparoscopic indications of fingerprints at crime scenes, as criminals are becoming increasingly knowledgeable, and the dissemination of criminalistics techniques in the media undoubtedly contributed to this. R. Fisher, an anthropologist, was the first to describe it in 1902, the biological phenomenon of arrangement of furrows on the lips. Edmond Locard, in the year 1932, of France, a criminologist, mentioned the use of lip print in identification of individuals. This was earlier suggested by Le Moyne Snyder in 1950 in a case where lip print was used to resolve the case. It was further suggested by Dr. Martins Santos in 1960 that lip features can be used in personal identification, and also gave classification of lip prints. A detailed study was done by Suzuki in the year 1967 on the measurements of lip prints. He used shades of lipsticks and techniques of its removal for obtaining valuable data for forensic application. Later in 1971, Suzuki and Tsushihashi conducted a study and they devised their own classification. In 1990, Kasperzak conducted research for the period of 5 years on 1500 persons to elaborate the practical use of lip prints. It was during the period of 2000 to 2012 that this study was carried out by several researchers from other countries and also in India. In 1967, Santos was the first person to categorize lip grooves into 4 categories. First, straight line, second, curved line, third, angled line, and fourth, sign-shaped line. Suzuki and Tsushihashi in 1970 developed a grouping method of lip prints. Suzuki and Tsushihashi classification gave first type, that is type 1, a clear cut groove running vertically across the lip. Type 1 dash, partial length groove of type 1. Type 2, a branched groove. Type 3, an intersected groove. Type 4, a reticular pattern, and type 5, other patterns. This organization is generally used for recording the pattern on the lips. Recording lip patterns. Lip patterns can be recorded in a number of ways. The suspect's lips are photographed. The lips are snapped, enlarged, and edge tracing of the furrows are created on a mirror. Lipstick is applied on the lips of the suspect or on the individual, and then they are asked to create an impression on a piece of paper or cellophane by pressing their lips on it. By application of a roller fingerprinter, by obtaining the impression of lips without the application of lipstick, and then processing it with the help of either conventional fingerprint developing powder or with a magna brush and magnetic powder. Mostly regular non-magnetic powders are used by various crime scene investigators, as it can be used on various surfaces like on windows, TV sets, etc. at burglary scenes. It can also be used on metal file cabinet or metal window frames. Various regular powders are available in market like black, silver grey, bichromatic and white. Appropriate color is selected to give appropriate contrast with the background surface in order to have clear picture of the latent print. Developing the latent lip prints. For recording of the lip prints by the magna brush method, the individual is asked to give an impression of his or her lips without the application of lipstick by pressing it on a glossy non-porous surface. The lip prints are subjected to hot air or allowed to air dry such that they solidify on drying. These prints are then developed with the help of magnetic powder and magna brush. Conventional powder methods are usually unsuitable for powdering lip prints, as the brush tends to smear or leave streak marks on the print. These streaks may be falsely interpreted by the comparer as the characteristics present on lips. As compared to the conventional powders, these magnetic powders and magna brush are very costly. Fingerprints and prints obtained from teeth are used as identification tool. Similarly, lip prints too can be employed as an identification tool. These prints are unique and do not change during an individual's lifetime. Prints can be found on surfaces such as on cutlery and crockery items, on the door or window glass, and on photograph or letters. Lip print may also be found on the edges or the sides of tooth mark on food items. In practice, lip prints have also been found on windows, painting, doors, plastic bags and cigarette butts. They are most frequently encountered in cases of murders, rapes and burglaries. Traces comprising of clear lines and unique elements permit individual identification of human beings. One may say that the lip prints are of same value as that of dactyloscopic traces. In the case of traces, in the shape of strains, the identification examination terminates with group identification. In their character, they are similar to other chemical and biological traces. Lip print identification is generally acceptable within the forensic science community as a means of positive identification because it appears in the literature. Lip print identification methodology, although seldom used, is very similar to fingerprint comparison and is known and accepted form of scientific comparison. There is no dissent in the forensic science community with regards to either the methodology used or fact that lip prints provide a positive identification. The Federal Bureau of Investigation, or the FBI, considers that lip prints are unique like fingerprints and are positive means of identification. The outer ear is constituted of a cartilaginous bone structure covered with teguments. The cartilage lamina is folded in on itself, forming protuberances and depressions that give the outer ear its characteristic shape, which has been only too well described in anatomical texts. Descriptions can also be found in the literature of the variations from the norm that can occur due to multiple causes such as abnormalities owing to disease, mechanical or surgical traumas or aesthetic reasons. Modifications caused by aging have also been described, relating to the increase in the laxity of the tissue, which manifests itself in an increase in the vertical length of the outer ear. There are furthermore significant variations due to ethnic origin, particularly with reference to the size of the ears. Diverse methods have been used to systematize the description of the outer ear. Bertillon made a purely descriptive analysis of its part. Schwalbe described the form of the helix, the lobe and 6 variants of Darwin's tubercle, besides the tragus, the antitragus and the angle of the outer ear to the brain. He designed 5 imaginary lines on the basis of which he spoke about the morphological index and the physiognomical index. Lana Rayleigh used a system of 4 lines centered in the tragus for the radial partition of the outer ear into 8 areas for comparative purposes. The ear print is a 2-dimensional reproduction of the parts of the outer ear that have touched a specific surface and that are usually the most prominent regions of the same, that is to say, are most commonly the helix, antihelix, tragus and antitragus. These types of prints, like fingerprints, are produced by the remains of desquamation, sweat and grease that the skin leaves on contact with the surface. They are not visible to the naked eye, but are recovered very easily with physical or chemical developers. The most common reason for the prints being left is simple. Criminals who are going to burgle a house lean their ears against the door to make sure that there is nobody inside and leave their print on it. The prints may be more or less fragmentary, marked or well defined depending on diverse factors such as the degree of greasiness of the skin, the application of the face or hair to the same area, or even the volume of sound that they were trying to hear. It is subsequently necessary to collect the print with a glass slide or methacrylate base. It has been reported that it is possible to determine approximately the height of the suspect by means of the ear print on the basis of floor to print distance with certain corrections. Once the latent print from the crime scene or the unknown print has been developed, we must compare it with a print of the ear of the suspect, that is a known print. In general, 3 methods have traditionally been used for the comparison of prints. First is superposition, that is placing one print over another with transparencies and comparing them. Direct comparison and dissection, dividing the print into sections and interchanging them to check the coincidences and superposition. The degree to which an ear print represents the original ear can be affected by diverse factors. The size itself can vary, as can the interdistances between the different points of reference depending on the degree of pressure exerted. The methods of print collection and analysis are designed to attempt to resolve these problems. Matte proposes a method of quantitative classification using a polar axis designed or drawn on the basis of a vertical line that is the tangent common to the internal margin of the impression of the antero-superior curvature of the helix and the tip of the tragus. Ingleby uses a similar geometric standardization, but claims that the definition of the polar axis proposed by Matt is difficult to employ in practice. Ingleby therefore calculates it with the use of a computer on the basis of centroids, that is, centers of intensity. In any case, the description of the inter-individual variations in the ear print represents a genuine challenge. The solution is outlined through image processing techniques with algorithms that permit us to draw from the print a reliable model of the characteristics of a specific outer ear. The forensic validity of the ear print is based on the possibility of identifying a particular ear as belonging to a particular subject. In practice, this can serve to rule out a subject as a suspect, to increase the evidence against a particular suspect, or even, if there are no suspects, to search for suspects in future databases. The limitations of the method are obvious and relate to the uniqueness of the ear print. Furthermore, it is possible from one single ear to leave different prints. This may be due to the manner in which the prints are made, principally, as has been mentioned, to the degree of pressure or the angle at which the ear was applied to the surface, but also to anatomical modifications of the outer ear. As has been mentioned earlier also, attempts have been made to deal with these intra-individual variations with diverse methods of analysis. The other problem is whether or not two different ears could leave a similar or identical print, or if, on the contrary, there is sufficient inter-individual variability to be able to distinguish between two different ears in all situations. The validity of the method depends fundamentally on the probability of two different ears leaving indistinguishable prints being reasonably small. In any case, the results of the analysis will be always expressed in terms of probability. Identification by means of ear printing is included amongst the so-called anthropomorphic tests. These are included among the body examinations aimed at identifying the accused. What is crucial is to determine the degree of significance that the said test should be given as a circumstantial evidence. Jurisprudence has accorded the status of probative evidence to the laparoscopic reports that prove indisputably that the prints found at the crime scene belong to the accused only. However, there is no jurisprudence nor doctrinal studies nor studies relating to comparative law on the value that can be given to an ear print found in a particular place. Legally, there is an important difference between whether the print does in fact irrefutably correspond to the ear of the accused, that is analogously to fingerprints, or if, on the contrary, it is a print that merely presents similar characteristics to those of the ear of the accused. It is hypothesized that all ears are different and that they present a series of characteristics that are capable of leaving prints that are valid for the investigation of certain crimes. Conceptually, the uniqueness of the ear is based on the so-called snowflake paradigm, that is, nature never repeats itself. However, it is clear that it is not possible to establish the individuality of the ear empirically, even though in practice it would be sufficient to be able to distinguish between any two ears through a finite number of characteristics. Despite the progress made over the last several years, the lack of support in scientific literature is the great weakness of the current state of ear printing identification. Ear and lip prints are important evidences just like that of fingerprints. Several parameters and classification has been proposed for correct characterization purposes and so as to help in positive identification. But these evidences are not fully entertained by the court of law. With all these information here, we come to the end of today's lecture. Do keep in mind what we discussed today. I'll be back with one more lecture in this series. If you want to learn more and enhance your knowledge, you may log on to our website www.cec.nic.in for MCQ, quizzes and LORs, etc. Thank you for your time today. I'll see you in the next lecture. Have a great day.